Thanks, Kenny. Look, when Kenny and I discussed this meeting on what time it would be, that was before the Eastern US went on a daylight saving and Australia went off daylight saving. So it's 8 o'clock on Wednesday morning here. So I think that means that I'm talking to you from the future. I thought I might start off by just showing you where I'm talking from. I'm in Tasmania, in Hobart, Tasmania. I'm just there. And like two of the speakers I've heard so far in these seminars, I'm also retired. I'm working from home. In fact, I'm talking to you from my study at home. And my home is just about a quarter of an hour out of the centre of the city of Hobart in southern Tasmania. And I'll just show you, this is the view. This is a little video I took from my study. So this is what I see if I look out the window. This one was actually taken a couple of years ago. There's a lot of wildlife around where I am, so this demonstrates I really am in Australia. But I'm here to talk about echidnas. When the echidnas were first described by Shaw in 1792, he thought they were clearly a connecting link between the two genera of porcupines, European porcupines, and the anteaters. And he put them in the genus Myrmecophaga. Again, when in 1800, when he published his General Zoology, he put the echidna together with the other anteaters. Now, where do you find echidnas in Australia? Well, pretty much everywhere. The density of echidnas there on this graph, on this graphic, shows more the number of people around to record them rather than the absence. So they're pretty well everywhere in Australia there are echidnas, but all the echidnas in Australia aren't the same. I should actually say something about long-beaked echidnas. First of all, would be saying a few words. The long-beaked echidnas are currently, confined to New Guinea. We've got western long-beaked echidnas, and right in the Bird's Head region, the Vogelkopf region, the east long-beaked echidnas, once again in the highlands, and amazingly, just a few months ago, Attenborough's long-beaked echidna was rediscovered, or video camera traps showed that it was still there in a fairly small area where it close to where it had previously been seen. This is Musa Opiang, who's done quite a lot of work on long-beaked echidnas. Is showing you a good picture of the scale. They are big animals with looking clearly like echidnas, but different echidnas. In Australia, there's five echidnas designated as subspecies of Tachyglossus aculeatus. Firstly, there's Lawsii up here, and particularly in this area in the tropics, and also New Guinea. This is the one that I'm really least sure about in terms of information and how closely, in fact, the mainland Australian one is related to the New Guinea one. But this is Lawsii. Tachyglossus aculeatus, the first one that was described, is on the east coast temperate zone. So these things are mostly... Ooh. Welcome, Catherine. And there's the Kangaroo Island echidna, just down on this little island off South Australia. Acantheon, the subspecies, covers the entire arid zone. And then there's the subspecies Setosis in Tasmania. And clearly there's, on the mainland, there are gradations between the various subspecies. So sometimes it's a bit hard to decide which one they are. But here's the Tasmanian echidna and a Kangaroo Island echidna. This is just a tracking transmitter antenna on it. And you can see what some of the differences between them. Here's a West Australian echidna researcher holding a Tasmanian echidna. Here's a Tasmanian echidna researcher holding a West Australian echidna, acanthion. This is the acanthion again at 104 days. Tasmanian echidna at 108 days, looking dramatically different for animals that are at the same age. But weaning in West Australian and subspecies acanthion is at 210 days, whereas in Tasmania, with cetosis, it's 150 days. Another picture of a Tasmanian echidna. Just look at the feet. These the claws on the hind feet, claws 2, claws 3. Echidnas use these back claws for combing, and this is clearly a comb. This one from northern New South Wales, you can see those 2 claws are very different. Once again, a very spiny animal, it needs a different sort of grooming device. So, xenarthrans and echidnas do share a few characteristics which makes them interesting. Well, it makes xenarthran interesting to echidna researchers, and I hope it makes echidnas interesting to xenarthran researchers. They have low body temperatures, they've got low metabolic rates, a very well developed olfactory system, simple teeth or no teeth, and the diet is largely insectivorous, apart from the sloths, which are herbivorous. And presumably mainly because of the low body temperature, the testes are internal. Looking at the diet, the armadillos, generalist insectivores, simple teeth, anteaters. These are the specialised ant and termite eaters. Also, they eat coleopterans, but they do eat, as far as I can read about it, they also eat coleopteran larvae. They have no teeth, they've got a long sticky tongue, and obvious comparison with the echidna, but Tamandua and the Cyclopes are both arboreal, so you would think the best comparisons would be with the giant anteater. But of course, they're much bigger than echidnas. Tachyglossus is only about 2 to 5 kilograms. The long-beaked echidnas, the glossus, maybe normally 4 to 9 kilograms, so some have been recorded up to 16 kilograms. And echidnas also have no teeth and a long sticky tongue, except I've asterisked the no teeth because actually echidnas do have a tooth. They have an egg tooth, which is a true tooth, but they need it to break their way out of the egg, and it disappears shortly afterwards. This is the Tasmanian echidna feeding it. If you watch the way it uses its beak, as we'll see, I'll show you later that it's a very flimsy looking structure when you look at the beak, but they use them very vigorously. And this is in sandy soil, and you see that it actually bulldozes its way through the soil. Interestingly, in West Australia, where there's a lot more sandy soil, the little trails that they leave may be about 10 metres or more long, just ploughing their way through the sand. Here's another Tasmanian echidna, this time eating ants in a log. You can see it's using its claws, front claws, and its beak to break up the log and get at the food. This is a North Queensland echidna, possibly Lawsii. Darker, but very, very hairy, and this termites will be an important part of their diet. You can actually see its back feet, once again, has a slightly different comb. And in this bit of video, you can see the use of the tongue. We will see the use of the tongue. Kangaroo Island echidna, multi-aculeatus, also, once again, quite a different animal. And it's worthwhile looking at the long-beaked echidna, I thought. You see the very different mode of feeding here, watching it probing much more gently, probing and pushing. And it strikes me that it's surprisingly similar to the method of feeding of a kiwi in New Zealand. Same probing. And in fact, the diets are very similar. So they're particularly feeding in leaf litter, eating worms in particular. So just a quick summary is that short-beaked echidnas eat ants, termites, scarab larvae, other soil invertebrates like crickets and moth larvae, but they very rarely seem to eat worms, whereas the long-beaked echidnas, worms are quite a big part of their diet. They eat scarab larvae, they eat crickets, they eat centipedes, but they definitely don't eat ants. So if I look at the diet of echidnas in Australia, in the arid and tropical zone, they're mostly eating ants and termites. When we get to the temperate zones, as we get more south and cooler, the number of termites decreases, and they start to have scarab larvae as an important part of their diet. Once again, here's another photo of the West Australian echidna subspecies, Acanthion, comparing with the Tasmanian echidna. And these are the skulls. This is the Acanthion skull and the Tasmanian skull. And there's a really massive difference between the relative lengths of the beak to the rest of the skull. So Tasmanian echidnas have got a much longer beak. And if we look at the West Australian subspecies in detail, if you look at the relationship between the length of the beak and the rainfall in the driest quarter, Western Australian echidnas from the driest areas have the shortest beaks. As it gets wetter, the beaks become bigger, but the overall skull size doesn't vary significantly. So in sandy, dry soils, there's more feeding on termites, there's more ploughing through the dirt, and for that, it seems that a shorter beak is more effective. So let's just have a look though. Here's the short-beaked echidna. This is the long-beaked echidna. And this is Megalibgulia, which used to be a fossil echidna. And the most recent of these, or the most recent fossil found, it was found in Tasmania, and it was only about 13,000 years ago that this echidna was walking around Tasmania. As they seem to disappear from the mainland a little bit earlier than this. But as the Australian continent dried out, it seems that the Megalibgulia and long-beaked echidnas were replaced by the short-beaked echidna. And you look at the Tachyglossus, the short-beaked echidna, you can see the beak is turned up, tends to be upturned, much better for this ploughing and pushing and vigorous activity we've seen on some of the videos, compared with the Zyglosus, which is that downturned beak, which is much better for probing. Megalibgulia, which has got a much more robust beak, and it's probably closer to the ancestral type. But clearly some of these three types coexisted, and short-beaked and echidnas and Megalibgulia have been found in the same fossil sites. So it seems likely that the ancestral echidnas ate a diet of invertebrates, not with much more variety than probably than the long-beaked echidnas. Anteating is often given as an example of convergence amongst mammal groups from different areas, and one of them, and this paper shows once again the echidnas, the anteaters. One of the key features is the reduced or absent teeth, which seem to be associated with anteating, but it seems quite, well, I think it's fairly clear that echidnas lost their teeth before they became anteaters. So it quite may be that anteating is one of the only niches open for toothless mammals in a fairly dry climate. So, and talking about the eating, how they eat, it's interesting paper was published by Peter Murray on echidna jaw mechanism now 40 years ago, more than 40 years ago, showing that echidnas don't open their jaws in a like hinge-like motion. They rotate the mandible. That means that they can take in new food while processing the last of the previous food that they've taken in. So they actually rotate the mandible and pull the food in with the tongue. And exactly the same sort of mechanism was later described in the giant anteater. Now, the reason I started working on echidnas, and because I was at the time working I was in a physiology department. I was very interested in temperature on organisms, and it was known that echidnas hibernate, and I wanted to investigate hibernation in Tasmanian echidnas. I wasn't particularly interested in things like diet, reproduction, but this thermal photographs show me holding an echidna, and you can see the temperature of the animals about 5 degrees. So this is a deep hibernating animal. So we tried in the lab, which is where we started, to investigate things like how the metabolism is depressed. This is a very physiological approach, but what we found was, although we got some interesting details on things like the control of respiration during hibernation, we couldn't really get animals to hibernate normally in captivity. And so the questions were raised then. What was the stimulus for hibernation? What was their normal pattern of hibernation? What actually were they doing? And the only way that we could answer these bigger questions was following and studying echidnas in the field. I won't go into some of the details of the field sites we've used, but I'll show you the hibernation period for males. So amazingly, this is a percentage of the males hibernating based on a fairly reasonable number. Male echidnas are going into hibernation in some of them going to hibernation in late January. Now, when I presented this at conferences, people would say, well, it's cold in Tasmania. No wonder they go into hibernation. In fact, it's not cold, particularly cold in Tasmania. And even if it were, late January is really the warmest time of the year. And we've shown that the echidnas are actually looking for the coolest part in their home range so that they can cool down and hibernate. And we see that starting to emerge from hibernation before the winter solstice. So they're coming out when it's even before the winter has reached its coldest. About the females, well, the females go into hibernation a bit later, and they come out of a hibernation later. And if we overlay them, we can see the difference between the two sexes. Now, those graphs include some juveniles, but they also, and you can see there's probably, this may be, this bit here in the males, probably a subset of males which are hibernated right through to quite to September. And there are also some juveniles which also tend to hibernate a bit longer. If we look at the changes in body mass in echidnas, this is one male that we followed over a few years, and I've just filled in its initial weight based on other data, but we first got him about here. But echidnas put on weight until their first hibernation. They tend to lose weight after they're weaned and then in their first hibernation, and then they come up to their maximum near adult weight in about 4 to 5 years. And we can see there's a big variation. There can be a big year-to-year variation. Echidnas have spurs, and I'll talk more about those later on. The first spurs, which are a mark of an adult male, we can observe in 2 to 3 years. The first mating, these are all matings from this male. You can see you've got 14 years of record here. The first mate, you can see matings occurring there, but he lost a lot of mass this year, in this particular year, and we thought he had died because we lost him, but we found him again, and he in fact reached a very high weight later on. And in the period when he was down at low mass, his body condition was poor, he wasn't involved in mating. And if we look at when the echidnas emerge from hibernation, when the males emerge from hibernation, the heavier they are, the earlier they emerge. So if the animal's in really good conditions, they come out of hibernation very early as May. If they're in poor condition, they come out a lot later, and they're much less likely to mate that year. The females, it's a different picture, and I've actually had to, this is shown with the date, emergence date here along the x-axis. Females that didn't mate hibernate a lot longer, and they are underweight. And these ones shown as having not mated, had mated in previous years. So it's not that they were too young to mate. But ones that are heavier, got more body fat, and emerging earlier. And I'll need to explain when you see some of the other data that this date of emergence isn't actually the date that they end their hibernation, but this date when they emerge isn't when they mate, but when they finally end their hibernation, because as we'll see, they can go back into hibernation after mating. Here's a graph showing body temperature and soil temperature of a male and female echidna from the same year. And you can see body temperature records, you can see it's reasonably, it varies over several degrees. And then this male has gone into hibernation in early February, and we see this typical hibernation pattern of cooling down. In this case, it's cooling right down and equilibrating with the soil temperature. And normally their body temperature in hibernation is about 0.5 degree above the soil temperature. So we've got this typical periodic arousals getting less frequent as the animal cools down. And then he's come out of hibernation in early June. And there we've got once again, the typical body temperature pattern for an echidna. For the female, she hasn't gone into hibernation. She goes into hibernation quite a bit later. And the pattern is not quite as clear, particularly here where it gets very messy during this arousal. Well, what's happened here is she was found mating. In fact, she was found mating with this male. So this male mated with this female. Mating has occurred here. And then she's gone back into hibernation for a month or so, come out of hibernation, And then she's dug a nursery burrow, moved into the nursery burrow. She's laid her egg here, and the body temperature is much less variable. Body temperature during this egg incubation and incubation of the young is very stable. The animal's in a burrow, but she also seemed to be physiologically regulating temperature much more closely. Also, I've got here the body weights, and you can see the male's losing weight during that hibernation period, and then he loses a bit more weight during the mating period, and then we get a big increase in weight during the spring period. The female, we've got loss of weight during the hibernation period, but also then during the feeding of the young. Now, the only xenarthran that's been known to hibernate is the pishi. And it's interesting to note that in this case, it's not deep body temperature, but it's subcutaneous temperature. But this is you look at this, you say, well, this is a very similar pattern to other hibernators, this cooling and the regular arousal. So it's interesting that the minimum subcutaneous temperature of the pishi was 14 degrees. The echidna minimum body temperature was 14 in the pishi, 4 degrees in the echidna. And what I think is interesting is the fact that we get an equilibration temperature, a minimum temperature that's above the temperature in the burrow, whereas the echidna is cooling right down till it's clearly equilibrated, but we're equilibrating a lot higher. Anyway, we took an echidna to the hospital and put it in the MRI machine to have a look inside, and you can see the testes are here, large, they're quite large, and they're sitting right against the kidneys. And the other thing that you can see, oh, let me just have, okay, so they're big. Percentage of body weight, the average is about something like 0.8% of the body weight is testes. Compare that with some of the other animals that are considered to be fairly promiscuous, rams, wallabies, humans. But also, if you look here, this is the brain, and you can see the brain looks fairly big, and you can see that it's, interestingly, the echidna brain is very heavily folded. As percentage of body weight, that is surprisingly large. And just digress onto this brain size for a moment. Here's a graph of body weight versus brain weight versus body weight for a number of mammals. Firstly, the eutherian mammals, which are shown in a selection of eutherian mammals, are shown in grey, and this regression line is the regression line for eutherian mammals. Okay. Not surprisingly, the primates sit well above it. Xenarthrans actually sit pretty close to this regression line. The marsupials tend to lie below it, and they actually get a bit more stupid the bigger they get. But the monotreme sit well outside the 95% confidence limits. That's the platypus, that's the short-beaked echidna, this is the long-beaked echidna. They do have very large very large brains for their body weights. Now, when we started looking at reproduction to work out the interaction between reproduction and hibernation, we started using ultrasound. They have internal testes. So one of my PhD students, Gemma, was trained by an veterinary ultrasonographer to use ultrasound for measuring things. And this is I think, a National Geographic film crew there. This lady was the journalist who's fascinated by echidna penises, but we actually won't get a chance to show those today. And I said the testes are up near the kidneys, and clearly she's not looking at doing the testes here. What she's looking at is the crural gland, because they have in the crural region, this is the gland, the crural gland, connected by a duct to this structure, the spurs. So echidnas have a spur. You might have heard about platypus spurs, but echidnas have spurs as well, and they produce a secretion. But interestingly, echidna spur is just floating there. The platypus spur is attached firmly to the bone near the ankle, but the echidna spur isn't attached, so they can't use it aggressively. And they probably are using it to spread some pheromonal or some substances which will perhaps help induce ovulation in the female, but certainly are involved with reproduction. Because here's a young in the burrow, 78 days old, this Tasmanian echidna at 78 days old. You see the little forming, there you go, the forming spur. This is a juvenile echidna just before weaning. You can see the spurs covered with the sheath. In the males, the sheath drops off and we get that rather sharp looking spur, but for females, the whole spur degenerates and we end up with just a spur pit, which has a sort of waxy exudate in it. But when we look at the difference between the testes in a non-breeding and breeding condition, this is the same animal in the non-breeding season and in the breeding season, you see there's massive change in testes size. And you can see one of the interesting things is that here's the testes start to increase in size before they go into hibernation. And so by the time the echidna emerges from hibernation, the testes are just about at their maximum volume. We have a period of about 2 to 3 weeks in which there's a little bit more development of the testes and spermatogenesis begins. And then this is the hibernation period. This is the reproductive period on average. And then after the mating season, the testes get smaller, shrink, and then once again we go through the cycle. One of the interesting things, well, there's two interesting things there, I suppose, is the fact that it grows before hibernation, which is unlike other things like ground squirrels, marmots, and so on, where the testes increase in size as they come out of hibernation, and that this growth is not testosterone dependent. Is testosterone here? Testosterone goes up, so it's up right at the start of the breeding period. But the crural gland also shows a big seasonal change, but that crural gland volume is clearly dependent on testosterone, and the crural gland obviously is it seems to be very important in male reproductive activity. So in males, there's very strong male-male competition, which leads to early mating, because we can see that the males in the best condition emerge from hibernation the earliest. And so if you're in very good condition, you can be out of hibernation early and you can mate before the other ones. And having the testes at maximum size does minimize the delay before sperm are produced. Echidna testes are so big and their metabolic rates are so low that it actually takes quite a long time to grow the testes. So they have to have them prepared before they enter hibernation. And speaking of sperm, this is echidna sperm. Oh, there's one drops off there. They look very, very reptilian rather than mammalian, and they form bundles. These sperm bundles are another indication of strong male-male competition. And this is quite amazing. Now this is a recording in vitro. In fact, the bundles are even bigger in life. So what we've seen so far places a problem on the females, because if they mate early, they're sure of getting the best males. But if they mate early, the young will be produced before the optimum time. The food availability will be low, and the mother has to lactate and feed the young. So the problem is, how does she do this? How does she mate with the best males but make sure the young are able to, she can feed the young. Well, she mates with those early males and then re-enters hibernation. But we'll see males then fight back against this. But hibernation appears to produce an embryonic diapause, and we get hibernation following by the females, as I've sort of shown in some of the diagrams, post-fertilization. And the longest that we've had this diapause is something like 50 days, because the female has stayed in hibernation and the development of the embryo just stops. Interestingly, compare it with the 9-banded armadillos, which also have a diapause, but this is caused by some means of ovarian regulation, hormonal regulation, which seems to be, which is the normal way that we see in other within mammals. So a quick run through the overall view of reproduction. In Tasmania, we have the gestation period after mating, assuming the animal doesn't go into hibernation, is about 21 days. The egg, she then lays the egg, and it is incubated in a pouch for about 10 days. It hatches, and then the young is incubated, continues to be incubated in the pouch and drink milk. And it's about 38 to 66 days. Typically about 6 weeks, the female leaves the young in the burrow and starts foraging. But we've got a long period here when the mother is in the burrow with the young, isn't leaving and feeding. So she's just running on her fat. She's been foraging beforehand, and then she goes out and foraging. And we've got a weaning at 150 days. But it varies around the country. In southeast Queensland, the gestation period is probably a couple of days, it appears to be a couple of days shorter. But the mother may forage with the young in the pouch, but when the young gets to a certain age, she then leaves it in the burrow because it's too big and too prickly to carry around. At Kangaroo Island, the mother may forage with the young in the pouch right from very early on, but we've got a much later weaning. So this, I think, illustrates what female echidnas would like to happen. We've got here an example where we've had and I should point out that we're measuring temperature just with a eye button glued to the animal's spine, so we're getting a skin temperature, but that's enough to tell us clearly when the animal's hibernating or not. So hibernating, and then in July she's mated, and we could tell by mating, if mating had occurred, by taking swabs from the female's cloaca, and we could see whether we had fresh sperm there or not. Okay, we had one mating, and we think there's another mating, which is when fertilization occurred, and a brief, some active period, and then she goes back into hibernation. And then we have the period of gestation. Progesterone comes up when she comes out of hibernation and peaks, as you'd expect, just before egg laying. You have egg laying, and then she's gone into, then she's in the nursery burrow with the young. So that's what the female would want: early mating, late egg laying, because she's able to hibernate while pregnant and stop the pregnancy, or slow the pregnancy, or freeze the pregnancy. But what mostly happens here, we have a female, mating, she's got pregnant, and then she's gone back into hibernation. Then some other males have found, and they hassle her. This is just repeated matings with various males, and then she gives up. She comes out to hibernate. She can't hibernate. She goes into the nursery burrow. She's laid her egg there, and we see this long period where the female is in the burrow with the young. This is a group of males pursuing a female. But that's something, perhaps, as you might expect, sometimes it doesn't work out very well. Here's a mating. And then she goes into hibernation, probably woken again or disturbed again by males, repeated males, harassment by males. And I think this female lost quite a bit of weight over this time. So she died in the nursery burrow after laying her egg. And here's another one where we've got mating, gone into hibernation with the young, come out because probably she's been found by a male. And then we have this repeated harassment by the males, lost in condition. She goes in the nursery burrow and dies. And I don't think this one, she even laid an egg. As a result of this, and as you would have noticed, the echidnas are quite long-lived. So its survival at 10 years for males is about 90%. At 17 years, it's about 64%. But the males live longer than the females. Some echidnas have been seen to survive more than 50 years. And that's only been males that's been observed in. But females got a higher mortality rate. They don't, and a lower survival than the males. So it looks like females expend more energy than males, and females can only normally wean one young every 4 years, and males breed. This, and I have to say, this is what we have observed in Tasmania. The breeding cycle hasn't been, reproduction hasn't been examined as closely in the field in other subspecies. The males breed 3 years and 4. On average, some years they just their condition isn't good enough, but peak energy expenditure for males and females occur at different times of the year. And interestingly, hibernation is an energy saving strategy, but it's not a necessity, because although all Tasmanian echidnas hibernate and all Australian echidnas show some degree of torpor during the same breeding period, so there are some out and feeding just about any time of the year. There's usually enough food around, but clearly an energy saving strategy, but you can't see that why they necessarily need to do it. But for the females, hibernation after fertilisation allows early mating, but egg laying can occur at the optimum time by using hibernation. Let's just look at some video that was filmed in Victoria, but it's essentially the same thing as happening in Tasmania, but it may be different as we go west in the mainland. Okay, so there's clearly a females being followed by one male, 2 males, 3 males. And I hadn't really noticed it before, but when I was preparing this for this talk, I had taken by the way that every now and again the echidnas just stop dead still. Sometimes it looks like they're probably doing some scent marking or something like that. There's one, that one's just stopped, one of the males. And he's done it again. So there's competition between the males. You couldn't say it was aggressive. One of the males has caught up with the female and he's managed to be copulating with her. The females stopped and the males haven't noticed. Oh, one of them really has realised. And this gives him a chance. Copulation again. And the other male realises what's happened and tries to break it up. So what we've found by taking swabs from the females, as I said, we can clearly tell if the females have mated, and you can, by looking at the smear of the sperm, you can tell how old it is. So you can see if the females mated several times, and we found that the females will mate many times with many different males. But interestingly, on Kangaroo Island, Peggy Rismiller, who's been working there for about the same period as I've been working in Tasmania on echidnas, though she hasn't done as invasive, that is, she hasn't actually checked things like the presence of sperm. Her observations suggest that the females only mate once. That's such a significant difference. I find it hard to be sure that could be true, and there's not more sort of cryptic mating going on. She also gets much longer periods of males following females, so males may follow on kangaroo and they follow the female for several weeks, or for many days at least. But she says the female only mates with one of them. In Tasmania, clearly, they tend to follow the female for only a much shorter time, but we get many mating. So the females and the males are both promiscuous. And interestingly, some clamiphorid armadillos show similar behaviour, though admittedly a little bit faster. We have, once again, males following the females, pursuing, nudging each other, and interestingly, sometimes they show this sort of same freezing behaviour. Okay, so I'll finish there with this drawing of a Tasmanian echidna drawn by one of my ex-PhD students. And
thank you, Stewart, for an excellent presentation on echidnas. And my name is Kenny Coogan. I am the education coordinator for the specialist group. And we have a very active website and social media. Our website is xenarthrans.org. And then we're on Instagram and Facebook. We post almost daily. And then if you go to our YouTube channel, you will see all of the past webinars and some animated videos, which we're going to talk about in a moment. If you go to our website, you'll see lots of different tabs. We have profiles on all 40 or so xenarthran species. And we have the only scientific journal dedicated to the conservation of xenarthrans, and this is on our website. But we have an exciting announcement. Our journal has been accepted into the Directory of Open Access Journal, DOAJ, which means lots of people, lots of more people can access our articles. And if you're interested in submitting an article for our journal, we can publish any time of the year, and the editorial process and how to submit an article is all available on our website. Earlier I mentioned that we have all 40 or so species listed. We have lots of statistics and data, and this is kind of for the scientist or the teacher to share. And then in the past year, what we did was we created a kids corner. We have 4 animated videos, one for anteaters, one for armadillos, one for sloths, and then one on the past, present, and future xenarthran. And all 4 of these videos are available in 3 languages, Portuguese, Spanish, and English. And to accompany each of these videos, we have coloring sheets and worksheets and mazes and lots of activities also available in those 3 languages that we would like you to utilize and pass around. And every time you're doing a xenarthran presentation, send them over to our website. If you have a pencil, get it handy. For this year, 2024, International Armadillo Day is always August 13th, so write it down. International Sloth Day is the 3rd Saturday in October. So that means the date changes every year. And World Anteater Day for the past couple of years has been on November 19th, and it's always going to be on November 19th. Coming up, this is not on our website or our social media yet, but in 2 days it will be. Coming soon, we have we do webinars monthly, but in June. We might have 2 webinars. So coming up, we are going to be talking about my favorite type of sloth, the maned sloths. We're going to be talking about silky anteaters, and we're going to be talking about xenarthrans of the Amazon, and we might have one more program, which we'll tease later. If you work with xenarthrans in a human care setting, or if you are a research scientist out in the wild and you have photos of them, or if you make a cake that looks like an armadillo, please send them to me so I can share them on our social media page. If you want to directly support xenarthran conservation, you can go to our website and there's a donate button on the top, available in all 3 languages, and you can donate money for equipment and conservation and education directly through PayPal, or I hope you can see it. I'm wearing my giant ground sloth t-shirt. You can go to our international store that drop ships, and you can get tank tops and t-shirts with all the different xenarthrans on there. We also have stickers, mugs, tote bags, and all of the proceeds of that goes to our conservation and education funds. And what really helps our education funds are our partner institutions via the Foundation for International Aid to Animals and Nurtured by Nature. All right, thank you. Stewart, do you see my ground sloth? I'm sorry I do not, my ground sloth t-shirt. I'm sorry I do not have an echidna t-shirt. All right.
Yes, and sorry, there's not a lot around. I don't know. I can't even offer you one because I don't have anything.
So, okay, Stuart, I'm going to read you a couple of questions. Okay, Mariella, which we can have come back maybe. She says, this is really fascinating information. How did you follow the echidnas over so many months to study their reproductive behavior in such detail? Did you equip them with radio transmitters? Are they reusing their burrows so you can easily find them in the field?
Yes, we did use tracking transmitters. Now, the best time to catch echidnas is for putting tracking transmitters on them is to look for them during the mating period. So once if you walk around the bush, as we did, we were working on a grazing property which had native vegetation in it as well. And the owner had said to us he had a lot of echidnas there, so we found some and put transmitters on them. And once you've got some, you're likely to find them during the mating period, because if you're tracking males, you're likely to find a female and other males. So this is the best time to get them is during the mating period. But we track them all the time, and we were just driving around looking for them. They're moving around slowly. Sometimes they're very hard to see. They're quite cryptic animals, because when we did sort of studies of the long term for some animals that would find and then not see them for a couple of years, and then find them again, and clearly they are in roughly the same place. So they're really often hard to find, but they turn up. So yes, we put transmitters on them, and to be confident, we really felt that we had to have about at least 12 animals with transmitters on them to have a good picture of what was going on. So we had 12, I think up to sometimes we'd have more, so something like 20 animals with transmitters on in our main field site. Interestingly, they don't reuse the burrows. In fact, when they hibernate, they don't build burrows. They just dig into the ground or into piles of leaves or bark, or just into clumps of tussocks of rushes and reeds. But so there's no burrow there. But when the females have young, they dig a nursery burrow, but animals didn't reuse the nursery burrows.
And you use the word cryptic a lot, and we always say xenarthrans are very cryptic, and that's why we need more funds to study them because they're so difficult to find in the first place.
We obviously need crypto coinage.
And Monique Poole, who did a webinar for us, she said, "Thank you. This was very interesting. And to see her presentation, head over to our YouTube page." And Justine asks, is there any sexual dimorphism in echidnas?
We have a paper on sexual dimorphism and developments, and looked for sexual dimorphism. And if you've got a large enough group, and you have to allow for the fact that there's such a massive seasonal variation within males and females, if you've got large enough, you can find a statistical difference with the males being slightly heavier than the females. But there's no way, if you picked up an animal and said this is a heavy animal, it must be a male or it must be a female. So there is a slight difference, but not really something you can use. And that we have the biggest data set, of course, the Tasmanian ones, and there seem to be slight differences between the subspecies, but that depends on the sample size they've used. So there isn't much. No, there is not in terms of size, but the way that we can tell, we can reliably tell if an echidna is a male or female, is to look for that presence of that spur. That spur is a giveaway, but that's quite difficult to find. You have to handle the echidna, pick it up, have a look, and when we're doing it, we're using anesthesia. We just say using isoflurane, so you could anesthetize them for about 2 or 3 minutes, enough, say, to check the sex, weigh them, and take a blood sample if necessary. But it's very difficult to tell unless you are an experienced echidna handler, really.
Mindy says, really fascinating. Thank you so much. Viva echidnas. And
Yes.
Jennifer, who's a wildlife care specialist at the San Diego Zoo Safari Park, says, we named a puggle after Stewart. Would love to tell him. And
so where was that? Sorry?
San Diego Zoo Safari Park.
Okay.
And
strange, I must say, Kenny, it's sort of strange. I haven't done a Zoom talk before, and it feels like you're talking into the void. And you're thinking, is there anybody there really? Have they all gone to sleep? What's going on? So it's better than not talking, but it's really interesting. More interesting if you can actually have live people there and see how people are reacting.
You had a good turnout.
Yes, well, that Kenya, is because I advertised on social media that you would send every person who attended a free sloth.
Yes. And Justine says, thank you. Very interesting. So similar to platypuses in regarding to telling the sexes apart.
Yes, except in platypuses, there's a big sexual dimorphism, massive sexual dimorphism. But they've got interesting similarities and differences with between the echidnas and platypus, and remembering. Oh, I realised that there was some slides I thought I had that were supposed to be there that disappeared, which just showed the ancestry of echidnas and when they first appeared. And so there's the latest ideas now are that the echidnas have diverged from a platypus-like ancestor more than 30 million years ago. Platypuses also don't have teeth, except juvenile platypuses have teeth, molars, which they lose on weaning when they start feeding. But in fact, we're doing a little paper on platypus jaw at the moment. But the platypuses, I mean, echidnas are particularly difficult to find any records on, just because they don't have teeth, and teeth are usually the things that are preserved. Oh, I was going to show you. Let me just show you an echidna. This is a skull, and you can see it's very flimsy looking, and this is the mandible. Now, when you look at this, think, how on earth do they poke so vigorously with this such a slight skull and such a tiny, very fragile mandible? So it's quite amazing when you see this. If you hold the skull up to the light, you can see through it, because it's very thin, because they've got virtually no muscles attached to the skull. Very muscle development is small because they're not using the normal jaw movements. But a very like a needle, that most unusual movement of the jaw means that you have obviously, well, there's no room for teeth, but platypus and echidnas clearly lost their teeth, their incisors, very early, but the echidnas have lost their molars as well.
All right, well, if Mariella, our chairperson, wants to unmute herself and join us. And I told Stewart that in 2027, the year 2027, I will be visiting him. And I went to Australia to study abroad 17 years ago, and I volunteered at a zoo. And for 6 months, and the first day at the zoo, you had to just rake up kangaroo poop. And I was quiet, and I did it because I wanted to be there for such a long time, and everybody else complained. And then about 2 hours before the end of the day, they came over and they said, "Kenny, would you like to come and feed the echidnas?" And I said, "Oh yes, I'd love to." And then they left everybody who complained there to rake up the kangaroo poop for the additional 2 hours of the day.
Well, nice to hear you'll be here in, as I said, in 2027. Now, I'll be 80 then, so don't book your travel plans too far in advance.
All right. Well, I'll be
I think
for it.
I need to do a sabbatical down there. I just love echidnas, and they're so similar to my peaches. So yes, I need to learn more about them.
It is interesting. I mean, that's why I've been watching this group, is how much echidnas have some of the characteristics that we see in various of the xenarthrans.
Right. It's really, I mean, in theory, they have nothing to do with each other, but still.
Yeah. Well, there's only a limited number of ways you can do things that work best, I guess.
Oh, there's an Echidna Keepers Facebook group.
I didn't know that.
Huh. I'll have to look that up. That's fun. Cool.
Well, and Adam is encouraging Stewart to join the Echidna Keepers Facebook group. So thank you, Stewart, and thank you for agreeing to do the presentation. And I look forward to the next 4 presentations. And if you know a xenarthran researcher, please reach out. And anything else, Mariella?
No, thank you so much, Stuart. I loved your talk. It's really fascinating.
Thank you, Mariella.
And yeah, looking forward to hearing more about your work or reading more about it.
So much to say.
Oh,
A little time.
Thank you, everybody, for joining us today.
Thank you.